So we still have a thrilling part to go, Arjen.
Absolutely.
So tell me what happened with the posters and with the people that were selected, and now we have some nominees here for the poster award.
Absolutely. So I'm not sure how we do this technically wise, but anyway, so what happened? Many of you were there yesterday and earlier in the week showing and looking at posters. Then we had this avatar event in Gathertown yesterday between 3:30 and 6:30. Then actually the university started to gain, because during the poster market there were actually 10 jury members that were incognito looking at your posters. And they selected the 10 best posters.
But did they have glasses and hats and beards?
You know, they had avatars anyway. So you probably met them, and they were your task force members in research programs.
We're going to ask them if they noticed, right?
I guess so.
Yeah.
And so then we selected the 4 bests from those 10. And so, of course, everybody is a winner, but we can only have 4 people here on stage. Actually, 3 people here on stage and one online, because he is abroad. And so the winners, the nominees for the poster awards are, and so this is Emma Coomans. Please come forward.
Yay, Emma. They will all have a table.
And then the second nominee is Lynn van Olst. She is actually going to be online.
Yes, on Zoom. Later. Yeah.
There she is.
There she is. Yeah.
Hi, Lynn. And then the third nominee is Aina Badia Soteras. We have seen her this morning, and she's back.
Special day for her.
And then the fourth nominee is Ana Millan Vidal. Please come forward.
They're ready. How we're going to do this is they'll be presenting their poster, maximum 5 minutes per person. And then after that, you will vote. So please listen to all of them and think about it. And then afterwards, you see in Slido a way to vote. Very easy, their name, and you vote for one of them. And then we will not show the results going up, etc. But when they're all in, we will declare the winner. So that's it. That's what we're going to do. You can tell which one, who will start.
Yes. So we will start with Emma. And the jury said about Emma is that she works on a unique population. So actually you work on Alzheimer's disease. I said, so it was excellently thought through. And the poster looked very, very, very clear. It was clearly presenting the problem and working towards the solution. And so that is, and I think the unique asset is that you work on homozygous twins. And so please take it away.
Thank you. Yeah, hi everyone. My name is Emma and I'm a PhD student from the Department of Radiology, Nuclear Medicine and the Alzheimer's Center Amsterdam. And I would like to show you some results of the work that I'm working on. My work focuses mainly on tau pathology in Alzheimer's disease, and we measure tau pathology by making PET scans. And PET imaging is a really cool imaging technique that allows us to not only measure how much tau pathology there is in the living human brain, but also where in the brain tau pathology is located. And we think tau pathology is super interesting to look into because it is very closely associated to cognitive symptoms and cognitive decline. And therefore also for prevention strategies, it is important to understand which factors contribute to tau pathology. Well, in this study we examined to what extent genetic and environmental factors contribute to tau pathology. And we did that using a quite unique approach, which is the monozygotic twin approach. Well, monozygotic twins, they are genetically identical, and therefore similarities between twins of the same pair can be due to genetic factors, but differences between twins of the same pair can only be attributed to environmental factors. So the results that you will see on this poster all focuses on within pair similarities in 2 characteristics of tau PET. First, tau load, so how much tau is there in the brain, and then second, tau spatial distribution, so where in the brain is tau located. So we performed tau PET in 78 twins, 39 pairs, and we first visually looked at all the images, and we observed that twin pairs were most often very similar in tau PET characteristics. And an example can be found in the bottom left, if I'm saying it correctly. In the top row, you can see 2 scans, and they show remarkable similarities in both the intensity and the regional distribution of tracer binding. However, there were also some twin pairs that were very different on tau PET, and one example is shown in the bottom row. You can see 2 twins again from the same pair, but one twin has much more tau pathology than his or her co-twin. Okay, so what we first did is we looked into similarities in tau load in identical twins. You can see that in figure 2 in the center at the top. And what we did for that is we correlated tau binding in a twin to tau binding in his or her co-twin. And if there's a strong genetic contribution, then twins will be very similar to each other, and this will result in a strong correlation across twin pairs. Well, we indeed observed a quite strong correlation across twin pairs for several brain regions, indicating that genetically identical twins are very similar in their amount of tau, and that genetic factors play an important role. What we then did in figure 3, that can be seen, is examined how similar twins are regarding their spatial distribution of tau pathology. And for that, we correlated each participant's spatial distribution of tau to that of every other participant. And this gives a measure of how similar a participant's spatial distribution is to that of his own co-twin, but also to that of every other participant in the cohort. And what we saw was that twins of the same twin pair were significantly more similar in their spatial distribution than twins of non-twin pairs, indicating also a role of genetic factors in the spatial distribution of tau. So what did we find? We found that genetically identical twins are very similar in tau load and in tau spatial distribution, indicating a very important role for genetic factors. But we also observed differences within some twin pairs, as can be seen in figure 1. And this indicates that there is a role for environmental factors as well. And it would be important to disentangle those environmental factors, as these can be potentially modifiable and thus interesting for future prevention strategies. Thank you so much.
Very good. Emma, Coomans. All right. So I think you took it off, and this is very. So what is the next step, Emma, in your project?
Yeah. So one of the next steps is to look into those environmental factors.
Yeah.
We looked into it a bit exploratory, but we want to look into it in a bit more detail as well. Something else is really to look into what the reason is behind this similarity in spatial location actually of tau, and whether there may be, whether there's a mechanism of connectivity that actually mediates this, so whether perhaps genetic factors influence connectivity patterns which influence tau patterns.
Very good.
And yeah, more look into that.
All right, we're looking forward to that.
Thanks.
Okay, so the next presenter is Lynn van Olst. So hi, Lynn.
Hello, can you hear me?
Yes, we can hear you. Where are you?
I'm in Austria. Very beautiful here.
All right. So
very sad to not be there, but...
So you were in Town yesterday, so you were actually with us, and you're still here now with us. The jury visited your poster, and I think the word that came up was that you had thrilling data. Not only, it was clearly presented, well thought through, very clear aims, visually very attractive, also I think a plus point. And then I think it was well discussed, but they said something extra is that you were actually, you had initiated the project yourself. So maybe we can look forward to how you did that and what your poster is about. Please, take it away.
Okay, cool. Hello everybody, greetings from Austria. I'm very sad to not be there, but it's very pretty here in the mountains. My name is Lynn. I'm a last year PhD student, and I'm very excited to pitch my research. And as you have seen today, people in our modern society are getting older and older, and resilience to aging has become a very important field of research. Increasing age is the most important risk factor for neurodegenerative diseases. And what is very important for my own research, aging is strongly associated with immunological changes. So during aging, the immune system becomes less efficient in mounting an effective immune response, and proteins that are released by immune cells, as for example, pro-inflammatory cytokines and chemokines, are increased in the blood of aged individuals, where they have a negative impact on brain function. For example, very interesting and complex animal studies have shown that you can rejuvenate the brain by exposing an old mice to young blood, and vice versa, that there is a loss of brain function in young mice when they were exposed to aged blood. So in this study, we hypothesized that the age-associated cognitive decline induced by age systemic factors were mediated via the immune system. So to investigate this, we first of all characterized all the immune cell types that are present in the normal aged mouse brain using single cell technology. And what might surprise you as a neuroresearcher is that there are many more other immune cells than only the brain resident microglia cells present in our brain. And it were exactly those other immune cells that we found to increasingly populate the aged mouse brain. And specifically, there were higher numbers of CD8 cytotoxic T lymphocytes. Also, if we looked into the T cells in the aged brain, they were less naive, which means that they had not yet recognized their antigen, while memory T cells in the aging mouse brain were increased, and these are cells that have already encountered and memorized their antigen. So then we asked if these age-associated systemic factors we talked about earlier were maybe able to induce immunological changes that we observed in aging. And to do so, we start giving systemic peripheral injections of old blood plasma to young mice. And what happened then was very exciting, because we found that those injections of old blood plasma into the periphery of young mice reduced naive T cells in the blood, same as we saw in the old animals. And when we zoomed into the brain, we found that there was one specific population of peripheral cells that was increased. And this is where it all comes together, because these were the CD8 cytotoxic T lymphocytes. And to infiltrate and crawl into the brain, immune cells will first have to bypass the blood-brain barrier. And at the blood-brain barrier, we found that specifically FECAN1, which is a molecule used by T lymphocytes to infiltrate the brain, was higher expressed after aged blood plasma. And we found that these higher levels of FECAM on the blood-brain barrier associated with higher numbers of CD8 cytotoxic T lymphocytes into the brain. So to sum this all up, our data shows the accumulation of memory T cells in the aged brain. And we show that systemic exposure to aged plasma in young mice can mimic this age-associated expansion of CD8 T cells in the brain parenchyma. And finally, I believe that this data opens potential ways through which maybe systemic modulation of CD8 T cells might increase resilience against brain aging in the future. So.
Good. This was Lynn van Olst.
Yeah.
Thank you, Lynn. So I'm going to go to the next round because we are a little bit short on time. So we have Aina again. So Aina, we know what how you are presenting, but we didn't look at your poster yet. So can you, like in a 3-minute pitch, take us through the essentials, and maybe also explain to me what the name aspirin comes from, and you know whether it would be itself a good therapeutic target.
Before you start, just take a look at this picture with all these women here. So we had to invite a man, otherwise it would be too many women, you know.
Yeah, female power.
Exactly.
This is Aina.
I cannot see the poster there, sorry.
Can you show the poster? It'll take it. Yeah. They'll do it. On the monitor, please. We still have Lynn there, also nice.
Yeah.
So I will try to summarize it in 3 minutes. So why we did this research is because there has been a lot of studies showing actually the signaling between astrocytes and neurons through exchange of glial transmitters and neurotransmitters, but very little studies actually tackle why the astrocytes and neurons are physically connected. So we wanted to investigate that in normal synaptic transmission and also in different memory processes. So to do that, we selected esrin, which is a protein that is enriched in these PAP processes, and we develop a knockout. To do that, we use the CRISPR-Cas9 gene editing technique, and then in the first figure, what we can see is that our virus tools are highly specific to targeting astrocytes, and they reduce the esrin protein levels around 75%. So then we took it further, and we also tried to analyze the morphology of the astrocyte. And we saw that this morphology also was reduced when esrin was lacking in the astrocytes. Then we zoom into the synaptic level, and we use two techniques. One technique that is a FRET-based technique, where we measure FRET between the astrocyte PAP and the presynaptic terminal, and the other one, the electron microscopy. So to make a summary of these both techniques, what we saw is that there was less synapses per astrocytic territory, and this probably was due to the fact that the astrocyte PAP was retracted from the synapse. Then to assess the physiological relevance of this manipulation, we measure basal synaptic transmission, and this was maintained. But what we realized that is if we would potentiate these synapses, so if we would stimulate these synapses, then we saw that actually glutamate was spilling over towards extracellular spaces. So we measured this glutamate spillover also using two techniques. One, a glutamate sensor that we express in the astrocytic processes, and we saw that indeed there was more glutamate in the perisynaptic space, and we also could link this exceed of glutamate spillover to a more activation of NMDA receptors. And finally, at the behavioral level, we also saw that this increased glutamate spillover and activation of NMDA also lead to an increased fear memory. So to wrap it up, I think what we can discuss about this project is that we are able to manipulate PAPs in vivo using esrin as a target, and that this de-engagement of the astrocyte PAP and the synapse leads to spillover and enhancement of a fear memory. And thank you so much.
Thank you very much. Okay, Anna, you are the fourth speaker. Our project is balancing off the preclinical work that we saw, and now you're going to go take us back to the clinic. You worked on modeling. You took computational modeling to the next level, looking at epileptic seizures spreading in networks, and that is what the jury admired in you. Please take it away.
Thank you. Thanks for the introduction and the great summary. So in this poster, I try to summarize the research that we are doing at the FU Medical Center, combining network science, computational physics with epilepsy research. In particular, we are looking at epilepsy surgery. This is the treatment of choice for patients with refractive epilepsy, so who continue to have seizures after being on medication. And it basically consists on removing the areas that are thought to be starting the seizures or propagating them, provided, of course, that this can be done safely and that they can be identified. So as you can imagine, there is a large battery of presurgical testing that is done before a patient undergoes surgery. This includes both structural and functional imaging, but still about 1/3 of the patients who undergo surgery continue to have seizures afterwards. So the goal here is to try to, using computational modeling, improve on these outcome rates. Now, the first practical problem that we need to face is, well, how are we going to model a seizure in the brain? Brain activity is not simple, so we really need to be careful on the model that we are going to define. The first the approach that we took was to actually simplify the picture and focus on the problem that we are trying to study, which is how ictal activity spreads on the brain from one region to another. And this is not that different to how any other spreading process takes place on a network. This has been characterized with epidemic spreading models, with which you are probably somewhat familiar after last year. And we can use that knowledge to our advantage here. So we defined a seizure spreading model based on epidemic spreading, And we use that to try different resections with the model, with the goal to gain better information before the surgery takes place. So the first question is, well, does the model work? Do the epidemics resemble seizures at all? That's the question that is answered in the first two panels there, where we compared the seizures in the patients. We can characterize them with depth electrode recordings, and which basically tell us the timing at which the seizure arrives at different brain regions. Then we can gain similar information in the model, so we can simulate a seizure or an epidemic using the the patient-specific brain connectivity. In this case, it was reconstructed with MEG data. And we also use other information from the specific patient, such as the area that was resected during the surgery or the hypothesized seizure onset zone. With that, we have a modeled seizure, and we can, again, find where the timing at which the seizure arrives at different brain regions, or nodes in this case. And we compare the two things, the one that we have modeled and the one recorded with the data. And we found, in this case, for instance, a 60% correlation between the model and the clinical recordings. And overall, in this group of patients, we found that we could reproduce, for all of them, the spatiotemporal patterns of seizure propagation. So even though this model is very simple and it neglects the underlying biological reality, it still seems to be capturing some of the key elements in seizure propagation. So, of course, we were quite happy about this, because it meant we could move forward with what we actually wanted to do, which is use the model to study epilepsy surgery. In the model, we can perform a resection by removing some nodes or disconnecting them from the rest of the network. And typically, what you will find by doing this is that the speed at which the seizure spreads decreases. So if before, at a given time, let's say 50% of the nodes were infected, now it's only going to be 40 or 30. And what you want to do is to find the resections as small as possible that decrease this speed of propagation as much as possible. And that's the bottom plot in the third column there, where we are looking at how the speed of the seizure decreases for resections of increasing size. The first finding is that for several patients, actually, small resections are already very effective. So maybe a partial resection that is less invasive than the one that took place could also be effective for them. But this is not true in all cases, because for some of the patients, a complete resection of the epidemic seed or the seizure onset zone was necessary to stop the spreading. So this is, again, pointing towards the need of personalized medicine. We really need to consider the underlying, the specific patient connectivity in order to find better resections tailored for each patient. We are now working on expanding the model so that it can suggest different resections in different brain areas. And we are moving towards the clinical applicability. So we are working on a pseudo-prospective study to analyze or test the applicability of the model. Thank you all for your attention. If you want more details, I direct you to our publications, which are listed on the paper. You can, of course, contact me. But I would like also to address you to our website, virtualsurgeon.net, which just went live, which is a portal where we try to bring the patients closer to the scientific research, which is also one of the goals at the core of this project. And thank you for your attention.
Well, they all did very well, so that makes it hard for you. Yes, please take out your phone, and at home or in the lab, or in your office, please think about it carefully. And then you have to make a choice. We will not show the... did you vote as well?
I was just going to see how it looks like.
Oh, it looks like
you're
going to be neutral, right?
So this is the second time that we saw, a complete panel of female power, like we showed 2 years ago when we did Pechacucha, also with 9 women actually. I'm sure there are many more people that could have stand here. And but these are 4 excellent nominees.
And all of them will win something, right?
They will all get a check.
Exactly.
They will all get this famous, this collector's item, the glass sculpture with the brain engraved.
Yeah.
They will all get flowers from a man.
Yes. Flowers from a man.
But actually, only the winner gets the main check of €400. So this is the exciting moment.
They can spend it.
They can spend it. They can spend it on anything they like.
Good night out. That's nice. So just keep voting and. With the delay, of course, there still will be votes coming in, and I will get a sign from Sibren, who's in the back, when the votes have stabilized, so that we know for sure. Yeah. But we're still not yet. Are you getting nervous already? How do you feel? And afterwards, of course, the winner can say something. We have here the flower ladies. Well, in fact, they have organized this whole event, so maybe we can thank them for now already. Thank you. Well, they did. Anita, Naomi. Lots of work, right?
And this is anyway a good moment to thank the organization, because this has been many weeks of work.
Yeah.
I thank all the technical parties. I thank my crew, the crew of Next Door's Neighbors Office. I thank Kim's, because she's been wonderful today.
Thank you. Thank you. People.
And I thank you for. I thank you for showing up and for you online to join us. And
people taking notes all morning long.
Very good.
In the top, yeah.
And also, while we wait for the results, I'd like to announce that we will be here. We will be back next year in this room. We're fighting still for the day. It has to be a day that Ajax is not on the soccer field, so we always have to struggle for a day. We are not the first priority in this stadium. But we will be back around the same time, same setting, so I look already forward to that.
Can you say it one more time like you said it the first time? We will be back.
We will be back.
That's lovely. Whoa, no. It's time. We have a winner. So what I'd say is you stand there so you can read it, and then it will be shown, and then you have to shout out the name. Okay, Arjen?
Absolutely.
You can do that? Okay, be a quiz master. There you go.
It says here, and the winner is, so I don't know yet. They don't know either.
They don't know.
And the winner is Lynn van Olst.
Woo!
Wonderful. So you should see. So you should see us, Lynn. They were all very excited for you. So I look at the camera. This is for you, Lynn. It comes with an award, and it comes with this, tremendous check of €400.
Yay,
congratulations.
Yeah. Thank you so
much. Then we have flowers. Flowers for all the other nominees. So you're also winners. There you go. There you go. Congratulations.
So Lynn, you can say something. You can cry or thank somebody, your parents or me or whatever.
Well, obviously the people that contributed to my study and my PI and all the lab members who were mental support as well, because I was kind of nervous. So a lot of people.
Congratulations.
Yeah. Thank you.
Wonderful. Lots of fun with the €400. Okay.
Thank you.
Thank you. Bye-bye.
All right.
Via Zoom, that's extra special, right?
Absolutely.
To do that.
Yeah, so she took that hurdle also.
And you also have enough to celebrate a good party, right?
We overloaded you with presents. Yes. That is what Diederik and I wanted. And on behalf of Diederik and the management team, I'd like to thank you all for showing up. And, let's continue doing this. Discuss the science, things that are close to our heart.
Yes, online you can still take a look at all the posters, and you can chat with each other. There's a private chat there, but also an open chat, so you can meet up, because we will be having lunch and talk to each other a little more. Yeah. Is there anything that you'd like to add or want to ask? Or look at that! Oh, we have the winners, and they all. Everybody gave the one they picked a one-word compliment, and the results are in now. Well, lovely, like exciting and awesome and great work.
I liked
cool.
the word wow. Very small.
Wow,
there.
yes.
It's wow.
And we have an emoji there. Maravilloso star, really clear, passionate. Well, these compliments are all yours. All right.
Very
so
good.
that's it. Anything else to add?
No, I think
Thanks.
I think we're done for it.
Right.
Yes.
Thank you all. Have a good lunch and see you. Bye-bye.
Bye-bye. Thank you very much.